Thank you very much, Dr. Elrod. Appreciate the opportunity to share this. This is a topic that's a lot of fun to discuss, and hopefully some people out there have some ideas or questions we can bounce around afterwards. Sorry. Okay. There we go. Okay. Yeah, so this, I guess I'll explain what we mean by nutrition 2.0 in a moment, but the whole idea is really that the field of nutrition has not instantaneously, but over the last few decades, gone through somewhat of a paradigm change that if we step back and look at it from a higher vantage point, I think can maybe open up some new ideas about how we can approach feeding livestock in the coming decades. So I guess starting off with the traditional way of viewing nutrition, if you really go back and look, nutrition didn't really become a true experimental science until really the 1850s. So it's not really that old of a science. And if you look back the first 100 years of this field, what really emerged is people kind of tried to assess what is it in the food that we eat or that animals eat that's critical to health growth. They basically came out with 3 reasons that we need nutrients. So nutrients either act as fuel, so they're energy sources. They can serve as building blocks. You can think here of amino acids or calcium for bone matrix. And then a lot of the micronutrients we consider being necessary only as cofactors. So most vitamins and some trace minerals primarily are helping to make enzymes active or help carry out a certain reaction. So in nutrition 1.0, the first 100 years in nutrition, this really was the paradigm around which we looked at nutrients being necessary or required in the diet. And I guess one way to think about that is it was a very, I mean, what we learned in that 100 years was incredibly useful and contributed greatly to huge increases in productivity across the livestock species, including dairy cattle. But I think it's important to sort of look ahead in the next 100 years and ask how much additional benefit can we get from looking at nutrition just in that way. And so this is kind of one of those futuristic thinking things that Mike Vanahar put together with some others a few years ago. And the point really here is to think about the inherent biological constraints of this system. So if we look at the lifetime energy efficiency of a dairy animal, and there's some assumptions here for sure, but across this scale, we're looking at cows that are eating at maintenance, in which case, at least in theory, they're not making any milk. They're only consuming enough calories to maintain body weight, to cows eating 2 times, 3 times, 4 times, and all the way up to 6 times that maintenance requirement. And so because of the dilution of that maintenance cost, which is 0% efficiency in terms of milk energy, going to 2x maintenance, and maybe you think the World War II era here, made a huge jump in lifetime energy efficiency. So even if you go from 1.5 to 2x maintenance, which you're still only at 33 pounds of milk per cow, that's a massive jump in how efficient you're using energy for milk. Then you look at what's happened in the last 50 years or so, and as you go from the 30, 40, 50 pounds of milk up towards the 80, 90, 100 pounds of milk, again, we're getting big gains going from maybe 25% energy efficiency. I'm sorry, I kind of said this wrong before. That 33 pounds equates to about 22% lifetime energy efficiency. Today, we're closer to 32 to 35% lifetime energy efficiency. But you can see that there's clearly diminishing returns as we increase milk yield and intake further. And there's some very logical reasons for this. So early on, we're diluting maintenance rapidly because most of the energy is going to maintenance. But by this point, we really are using about 3/4 of the energy in a lactating cow for milk. And so the marginal benefit of shrinking that further becomes less and less. Furthermore, at some point, when we start getting nutrients flowing through the animal extremely quickly, particularly since we have to feed fiber to ruminants, we are going to lose digestibility at some point. So the point of this really is we've made huge gains in the last 100 years, many of them due to dilution and maintenance. And that gravy train is almost done. Just because we've made so much progress, we are hitting the point of diminishing returns. Okay, so the question is, you know, what do we do from here? Do we just give up on additional advancements, or have we met our potential? And I think the answer is no, but I think we have to think about things in a different way to get additional improvements in efficiency. Okay, so I'll circle back to that later. So one thing that gives me hope in that respect and makes me think we don't have to give up on further efficiency gains is that we have a lot more things that nutritionists think about today. Okay, so again, in Nutrition 1.0, perhaps you could phrase it this way. We asked, what are the levels of nutrients that we need to maintain normal health and support good productivity? And we thought about that in terms of meeting requirements for fuels, for building blocks, and for those cofactors. But in this new paradigm, we certainly don't throw that out at all. Of course, we need all those things. But beyond that now, I'd argue that we can think about whether nutrients can confer robustness or resilience, if you prefer that term. Okay, so I'll explain why I think that's a legitimate question through this presentation. Part of what drives that new paradigm is really just new discoveries. And there's 5 of them that we identified in a review paper in 2016 that, you know, I took this image from. And some of these are not really as new, particularly in the ruminant world, and we'll walk through these. But really all of them open up new avenues by which nutrients can influence animal health and productivity that, you know, 50 years ago, at least most of the nutrition world would not have considered a possibility, or at least a primary focus. So starting off with host microbe leaks. So this is an area where nutritionists in the ruminant world would kind of stand out as having figured this out or focused on this far more than human nutritionists or monogastric nutritionists, simply because you couldn't avoid the conclusion that part of what we're doing in feeding ruminants is altering the microbiota in the rumen, and it had very clear effects. So this is less new in our world, but still, if you go back to that paradigm I laid out, maybe overly simplistically, but in Nutrition 1.0, the way we thought about nutrients is delivering those nutrients to the animal couldn't account for some of the microbe-mediated effects that we see today. So 3 big areas where this can work. One, obviously, nutrients are providing substrate not only for the host, but also for the gut microbiota. And so that's going to have effects. There are some nutrients that are inhibitory to gut microbes. Perhaps the most obvious, if you have any interest in swine nutrition, would be the high levels of zinc that we can feed and have massive effects on the hindgut microbiome. And then something that's relatively new, to me at least, is this concept that the gut itself can release nutrients to sort of culture its own microbiome. There's some really neat evidence that during disease events, the gut actually shifts the oligosaccharides that it's releasing to sort of try to prop up the healthy gut microbes. Okay, but I'm really just going to focus on number 1 here for the sake of time. And again, lay out some maybe obvious things, but again, stepping back and looking from a higher vantage point, just making the explicit point that this is not fitting in Nutrition 1.0. So this is data from a huge data set, 1,932 observations of dairy cattle fed different diets. And the conclusion from this regression is that increasing dietary starch is associated with a decrease in total tract NDF digestibility. And I suspect that surprises nobody on this call. But again, the point being, in Nutrition 1.0, there was no allowance for nutrients to alter digestibility of other nutrients. Okay, short of maybe a toxic effect, which I would argue this is probably doesn't really fit in that concept very well. Okay, so how does this happen? Well, I think probably we'd all agree that it's most likely the increase in starch alters the rumen environment such that fiber digesting bacteria are less competitive and become somewhat decreased in terms of a percentage of the microbiome there. And as a result, you have less production of fiber digesting enzymes and a decrease in fiber digestibility. Okay, so it's one of the things, again, in rumen nutrition we're used to thinking about, but it is kind of a novel concept to the field as a whole. So I'm not going to spend as much time on this. Everyone here is familiar with this concept, but nutrients affect digestibility of themselves or other nutrients through the gut microbiota. And what we are learning more from the monogastric world, but microbes that are influenced by the diet can then in turn affect the gut tissue and maintenance costs. So they can either have positive or negative effects. Volatile fatty acid production in the rumen and the hindgut, particularly butyrate, provides a key energy source for those epithelial cells. Conversely, if we allow for proliferation of pathobionts, microbes that are typically there but can become pathogenic, they can cause great harm to the gut and massively increase the maintenance costs by just inducing a immune response to heal that or protect from that damage. Okay. So big effects through the microbiome. Another concept that's emerged and become very solid, but is maybe less familiar to rumen nutritionists, is this idea that nutrients don't just serve as building blocks, cofactors, and fuels, but can also serve as signals. So essentially, acting in a way that 20 years ago we thought only hormones could act. And shown in this image is the idea that nutrients can serve either as a ligand for receptors on the cell membrane or for nuclear receptors in the interacting with DNA. And the list of nutrients that can act in this way has become almost as long as the list of nutrients. When this first emerged, it was largely focused on long-chain fatty acids working through nuclear receptors. And then the human genome was sequenced, and they discovered, to their surprise, that this little class of membrane receptors that looked like rhodopsin, the receptor for light in the eyeballs, actually had something like 700 members, and the vast majority of which people had no idea what they did. And we found some of them were hormone receptors, but many of them by now, if we know what they do, some of them we don't, many of them are actually known now to be receptors for nutrients. And so the list of these G protein receptors, what their ligands are include short chain fatty acids, sugars, amino acids, some minerals even, which really surprised me, and then also metabolites produced in the body. So these include ketone bodies, lactate, and many others. So it really opens up a wide variety of possible responses to diet that we really hadn't considered before the early 1990s. And really, this is still an emerging field. So there are still labs and biochemistry departments around the world sort of working on understanding what several hundred of these G protein receptors do. So let's think about some implications. I mean, that's very biochemistry. You know, what could that mean at the whole animal level? Well, something we talk about a lot in the dairy world today is the amino acid, balancing for amino acids, meeting those requirements for not just metabolizable protein, but metabolizable methionine, lysine, arginine, etc. And I know I was taught, and probably most of the people on this call were taught, this sort of rain barrel concept of amino acid requirements. So, and there's a very good biochemistry underlying to this, this concept is that if you have a DNA sequence that calls for a methionine followed by a valine, followed by a leucine, followed by tryptophan, that sequence of a protein cannot be changed. And therefore, if you're short on even one of those amino acids called for in that protein can't be made. Therefore, for the essential amino acids that cannot be synthesized in the body, you need a certain amount of each one of those amino acids. And if you express those as a percent of the established requirement, the one that is the shortest supply will constrain whole body protein synthesis, which presumably in a dairy cow would be seen in terms of decreased milk protein yield. So this rain barrel concept kind of makes that point. You can only have so much water in that barrel, and it's driven by the shortest stave, and so you need to address that limitation. Okay. Well, how much experimental evidence is there that this is actually true? And in ruminants, it's actually quite difficult to do these studies. So part of the reason we don't have a lot of data is it's very difficult research to do. But let's talk through one experiment that at least kind of draws that into question. Okay, so this is from Mark Hannigan's group at Virginia Tech, and this is using mammary tissue in the culture plates. Okay, so it's not in the animal, partly because again these studies would be very difficult and expensive to do in the animal. But so take it with a grain of salt, if you will. But this is using mammary tissue in culture and looking at casein fractional synthesis rate. That's CFSR, casein fractional synthesis rate. So it's saying how much casein can this tissue make per hour. And starting off, they added 4 essential amino acids that they thought could be limiting this system. They added them together. So isoleucine, leucine, methionine, and threonine added in combination, and very clearly observed about a 4-fold increase, a maximum of 4-fold increase in casein synthesis, pretty clearly responding to that set of amino acids. Okay, there's nothing about this that disagrees with the rain barrel model. Any one of those 4 amino acids could have been limiting. In fact, perhaps more than one was limiting at some point. It was sort of co-limiting, or there was a second limiting one as well, and so it would have addressed those issues. Okay. The problem comes in the next experiment. So here they split out those 4 essentials and added them independently with the same basal media. And you can see to varying degrees, each one of these 4 amino acids independently increased casein synthesis rate. Okay, so if you buy this in vitro model and you take this to the rain barrel concept, this would say that each of these 4 amino acids was the shortest stave, which obviously just doesn't make sense. The odds of all 4 of them being equally co-limiting somehow, doesn't make sense. Plus, even if they are co-limiting, you should need to add all 4 of them simultaneously to see, an increase in the water in the rain barrel. And if you really want to dig in and be critical of that model, you can find similar data that's just doesn't fit with the concept, and lots of monogastric data. Okay. So really, and mea culpa, I still teach the rain barrel model to undergraduates because I haven't really figured out exactly at a sophomore undergraduate level how to explain this in a clear way, because we haven't had a crystallized replacement of it emerge. But we do have some biochemistry that would fit with a more complex model. Okay, and a lot of the literature in protein signaling today will talk about mTOR, which stands for mammalian target of rapamycin complex. So it's a 5-protein complex. It's named after this drug rapamycin that has huge impacts on growth. But what's interesting about the TORC complex is it responds not only to amino acids in a way that we don't really understand, but it also can be influenced by oxygen supply, by energy supply, by hormones. And it really, thinking about protein synthesis through this signaling process really does help to explain some observations that have been made over time. So, for example, in some of these same studies, like I showed you from Mark Hannigan's group, where they show responses to amino acids, they're also able to show that you can increase protein synthesis by supplying more energy, even in a system that's probably not starved for energy, where it's not truly limiting. And they make a pretty good argument that that's probably through this same signaling complex that's integrating all these different signals. So in this model, again, where it fits with past data is if you're badly deficient in methionine or lysine, the mTOR complex will sense that and will slow down activation of protein synthesis machinery to decrease that translation of proteins, because you simply don't have the the building blocks to carry that out successfully. However, it's not necessarily the case that if methionine is somewhat limiting and you have just sufficient lysine, that this complex could not potentially respond to additional lysine. And that's where it differs from the rain barrel complex. So again, if we think about this traditional model where here you'd say, lysine's the only amino acid that you could increase protein synthesis with, some of these ideas may be turning that barrel on its side. So maybe that's not as true and maybe not as black and white as we used to think. Okay, moving on to the next concept. One thing that my group's been very interested in is this idea that nutrients really interact with the immune system a lot. And not just that, but metabolic tissues, organs that we think of as primarily being metabolic, actually interact in a really dramatic way, and not just during disease, with the immune system as well. So I'll just give you a couple examples. We could spend a whole hour just on that. But here's one example that's really relevant to dairy cows. A really nice paper from a decade ago now showing that if you take neutrophils out of dairy cows, isolate them and so that you can test them in cell culture, and then incubate them with increasing amounts of ketone bodies, BHBA, you can show a quite dramatic decrease in a couple critical functions of those cells. So on the left here, it shows the proportion of cells that could effectively kill a bacteria after engulfing them, which was not harmed in this experiment. But also another key part of that, which is phagocytosis. So that's where the neutrophil engulfs the bacteria so that it can destroy it inside the cell. You can see that although these cells were very capable of killing bacteria, they didn't carry out the first step very well when they were incubated with ketone bodies. So even at 1 millimolar BHBA, which would be below the threshold for subclinical ketosis, there's a 50% reduction in the proportion of those cells that were engulfing E. coli as a model bacteria. Okay, so really dramatic effect there. Secondly, on the right, there's a newer mechanism, well, a more newly discovered mechanism by which neutrophils can kill bacteria, and that's called extracellular killing. This is, I always compare it to Spider-Man, that the cell actually commits suicide, releases its DNA in a sticky web, and actually traps bacteria in that web, and it somehow leads to their death. Again, incubating with ketone bodies, even at very subclinical levels, led to a pretty dramatic drop in the proportion of cells going through that process. So both of the two main killing mechanisms by which neutrophils eliminate bacteria were pretty dramatically inhibited by BHBA in this experiment. Okay, so a very clear mechanism by which a nutrient can alter an immune function, and scaling up to the whole animal level, then providing a mechanism by which we can maybe explain the epidemiological links between ketosis and subsequent mastitis, metritis, and other infections that we deal with in transition cows. So is this a unique thing with ketone bodies? No, I don't think so. Like I said, we could show lots of data like that. And this is a really interesting review from about 13 years ago now. And in this review, they point out something that I was not aware of before looking at it as someone who's not really focused on fly biology, pointing out that insects, and using Drosophila here as an example, have an organ called a fat body. And apparently the fat body actually serves a combined role for the fly that basically comprises all the things that we expect our liver, adipose tissue, and the entire immune system to do. And what's unique about the fat body is all those cell types are sitting side by side and carrying out a lot of crosstalk, communicating, signaling to each other, directly interacting. And the point of this comparison is really to say, look, over 600 million years of evolution, these functions have been geographically segregated, but what we're learning today is the signals that are going between those cell types in the fat body, those same signals somewhat evolved, but the same signals are essentially working throughout the system in us as well. So there's still now crosstalk between adipose tissue and liver that we've really just discovered since the Human Genome Project, and far more interactions between resident immune cells in the liver and adipose tissue and in the blood with these metabolic organs than we used to realize. Okay, and that's changing. Again, it's changing the way we think about the immune system. It's changing the way we think about metabolic control. Nutrigenomics is a pretty broad term, and I get frustrated sometimes because people use it probably in many different ways that mean different things. I'm going to use it today to talk about really two somewhat unique interactions between genetics and nutrition. The first being the idea that genetic variation, here I'm showing a single nucleotide polymorphism, a SNP, influencing how nutrients are used. Okay, I want to talk about an example of that. This is a study from the feedlot world, but the lead author is Paul Kononov, a dairy nutritionist at the University of Nebraska. Probably the best example I'm aware of in the livestock world of a really, really clear genetic interaction with a management tool. Okay, so here they're either feeding zilpaterol, a beta agonist that increases growth efficiency and muscle mass in the late stages of the feeding period for feedlot cattle, showing that interacting with a single nucleotide polymorphism in the leptin gene. Okay, so step back 1 second. If you're not familiar with leptin, this is a hormone produced by adipose tissue that leads to a suppression of appetite. So the idea is here, as an animal increases in adiposity, has more and more body fat, more leptin is produced, telling the animal that it's sufficiently fat that it should back off on its feed intake. Okay. So we've known for some time in cattle that there's this really impactful genetic variation at a certain place in that gene, where sort of considered the CC genotype is considered normal, and you can either have a heterozygote or a homozygote switch to a TT genotype there. Okay. So dry matter intake was an outcome of interest here simply because leptin is known to alter appetite. You can see that on average, the normal genotype ate about 9.7 kilograms per day, regardless of whether they got the beta agonist or not. But as you introduce that genetic variation, you can see that there's a huge interaction with zilpaterol, where the mutant, if you will, the TT genotype, shows about a 5% drop in feed intake if zilpaterol was fed, whereas if they were not fed zilpaterol, they showed about a 5% increase in feed intake. Okay, and that can be good or bad, right? Because if you can get the same growth from those animals with less intake, maybe that's what you want. But what they found is those TT genotypes fed zilpaterol showed a 33% drop in choice carcasses. Okay. Now today, that's not as expensive of a fall off as it would have been a decade ago, but still, that's a huge effect. And this was a big enough deal that this work was done in a very large commercial feeding business that has many lots. And at least for some time, I understand that they were actually genotyping most of their animals coming into their lots to actually make decisions about whether they were going to use the beta agonist or not. So that tells you, what they thought about this interaction, how impactful it would be. Okay, so switch now to thinking about how many of these SNPs or other genetic variations exist in the dairy cattle population, and how many different nutritional strategies we use that could be influenced by the underlying genotype. We don't even really know about differential effects of nutrition in Holsteins versus Jerseys, entire breeds, much less individual genotypic differences in response within a breed. So there's kind of an unlimited amount of digging we could do here. Secondly, I want to look at another potential nutrition genetic interaction, and this is more to the concept that nutrients can actually influence the chemistry of DNA, if you will, and therefore influence what genes are actually expressed. And let me explain that a little bit. So this field is known as epigenetics, and epigenetics really refers to the idea, well, epi meaning above, I think, basically what it means, above genetics, layered on top of the DNA. The concept would say that, yes, you and I or our animals are inheriting many of our traits from parents based on the DNA sequence that you inherit. However, how that DNA is expressed can be altered dramatically by environmental influences on, again, the chemistry of DNA. And a couple of the really key chemical modifications of DNA that influence expression of that DNA would be modification of histone proteins, which are sort of the spindles that allow DNA to be packed tightly in a chromosome. And so those spindles can be unwound or wound up tightly, which is going to influence how much that DNA can be accessed for expression. And then at the more micro level, the DNA itself can have chemical modifications. And this is not the only one, but the main one people think about is methylation. So methyl group added to certain bases can make it easier or harder for well, transcription factors to interact with that DNA and start the process of a gene being expressed. So again, lots of environmental influences on these things. There's big papers now showing that exposure to secondhand smoke, for example, can change some of these methylation patterns, for example. And what's really driven a lot of interest in this is there's some evidence that some of these patterns stay somewhat constant even throughout a lifetime. So that in theory, parents can increase or decrease exposure to different things, alter methylation of the DNA of their offspring, and that could influence the entire lifespan of that offspring. Okay, so how real is this in the livestock world? Again, I'm just going to show what I think is the most interesting study I've seen. We're kind of at the early phase of this in the livestock world. But in poultry, they have some advantage in that the animals are relatively low cost, the lifespan is relatively short. And this is a study out of Australia, where in Australia, they don't produce a lot of their own grains. So people in the monogastric world tend to switch between wheat and corn based on import prices for those different grains. And so it can, within a year, they can be switching back and forth between different sources. So they asked this very fundamental and actually applicable question for the poultry industry in Australia of, okay, if we feed layer hens a corn-based diet versus a wheat-based diet, how is that going to influence the broilers then that we produce from those layers? I said that wrong. I'm sorry. I'm not used to talking about poultry. Layers are for making eggs, right? So these are the broiler breeders producing broiler chicks. And so, okay, so they're testing that. What if we leave the next generation on the same grain type? But then they said, alternatively, what if we switch? So what if we take the hens on wheat and feed the broiler offspring on corn and vice versa? Okay, so this was pretty shocking to me. I would have thought that would have had almost no effect at all. But when they looked at body weight of the chicks in the first 5 weeks of life, you could see, regardless of what grain source they were looking at, so this wasn't a wheat versus corn advantage or disadvantage. Basically, whether they went corn or wheat, staying on the same grain led to better growth than switching either direction, and which is why they're showing it sort of pooled form here. Okay, so that's pretty interesting. But beyond that, this is kind of amazing. The mortality rate when they swapped grains doubled. So for somehow, and they have no idea what exactly the genetic architecture of this is, or epigenetic, I should say, but for some reason, when the hens were adapted to one type of grain, they were able to pass on some kind of signal to the offspring that allowed them to perform better and be healthier by staying on that same type of grain. It's amazing to me. Okay, so we are starting to get into some interesting data on this in the dairy world. There is some work that's been done now showing that sort of gestational feeding of some products seems to be influencing calf trajectories from there, and it's really just starting to become a topic. And we don't really know, even for those examples that are pretty clear, again, we don't really know at the DNA level exactly what's going on, but we at least have some inkling that it has something to do with this epigenetics phenomenon. Okay, so if you encompass all these sort of new ways of understanding nutrition, how does that really impact the way we think about nutrients? Okay, so let me go through butyrate as just one humble example of this. So butyrate is obviously a volatile fatty acid produced in the rumen, produced in the hindgut as well. And I would say in Nutrition 1.0, we thought about this maybe in two key ways. One, it's a fuel, right? We think about, okay, you put it in your model as 0.51 megacals per mole of butyrate. And then also it can serve as a building block because you can turn that into a fatty acid in milk or what have you, okay? So pretty straightforward, okay? How does that differ? Well, now, for starters, we have at least two, and I would argue at least three receptors that actually respond to butyrate. So these are cell membrane receptors that are sensing butyrate and changing their function in response to that signal. And the cells that respond to that include enteroendocrine cells. So these are hormone-producing cells in the gut, making gut peptides and signaling to the rest of the body. So through that mechanism, butyrate can influence function of the rest of the body. It's being sensed by nerve cells in the gut. It's also being sensed by leukocytes, by immune cells that are found in the gut. Okay. So lots of potential whole body implications of butyrate as a signal. Two, butyrate's also in this epigenetics game. Okay, there are a series of proteins called histone deacetylases, histone acetyltransferases that are adding or subtracting acetyl modifications to lysines on histones, which are, again, the proteins that we wrap DNA around. And in general, more acetylation leads to a more active region of DNA, whereas deacetylation tends to silence that DNA. Well, butyrate is actually one of the most potent inhibitors of histone deacetylases, and thereby acting to keep DNA in a more active state compared to the absence of butyrate. So again, changing this epigenetic mechanism of making DNA more or less available for expression. Thirdly, butyrate plays important roles simply as a fuel. So we already talked about Nutrition 1.0, yes, it's a fuel, but what wasn't really accounted for in that more simplistic model is even in an animal that's not starving, the presence of a fuel at the right kind of fuel in the right place in the right time can actually change tissue health. And so this is from a really intensive study where they were asking, why does butyrate lead to more healthy colonocyte function? So the epithelial cells in the colon, there's some evidence that butyrate is anti-carcinogenic. Clearly it improves health of that tissue. Why is what they were asking. Well, they said it wasn't, they wondered if it was through those receptors, G protein receptors. They wondered if it was through epigenetics. But what they ended up deciding is the main benefit here was really by preventing cell death. And it's a fancy form of cell death called autophagy that butyrate inhibited simply by providing a fuel. And again, this was not cells that were completely starved, but it's such an important fuel for those cells that it keeps them alive. And so it's changing tissue morphology even. Okay. So if you add up all these sort of physiological effects and look at a real in vivo ruminant study, you can actually see some responses to this. So this is feeding sodium butyrate in starter grain for just 4 or 14 days at the beginning of life in calves. And they were able to show an increase in ruminal epithelial thickness, showing growth in that tissue early in life. So again, in butyrate, for example, nutrition 1.0, pretty simple effects, but now we think it's a receptor ligand. It's altering DNA accessibility. It's a fuel still, but it's basically altering physiology beyond just providing those simple roles that it played in our first paradigm. All right, so the problem with this paradigm is that's one metabolite. We have lots of mechanisms to think through. Now, if you apply that by all the metabolites made by microbes, made by the host, all the nutrients coming in, and this becomes really pie in the sky stuff. There's almost nothing that you can't come up with some pathway to justify some, crazy claim. All right. So how do we turn this science into relevant information that we can use on farms? Well, I think starting out with, we need to at least consider different outcomes that we maybe didn't before. So I would say this opens up opportunities for very long-term effects of nutrients. So it is possible, and I think we can point to a few rare examples where provision of a nutrient is going to influence the next generation even 3 or 4 years later. Well, and that animal itself as well. Okay. It makes it likely that these responses vary between physiological states. So for example, if a nutrient's having an important effect by binding to a receptor, if that receptor is up or down regulated at different phases of life, then the response to that nutrient can be very different. It highlights a lot of potential dietary interactions through the microbiome, for example, but also through, let's say, nutrient A turns on a receptor that then responds to nutrient B. There are lots of those types of interactions that didn't really fit in nutrition 1.0. And lastly, because of this pie in the sky sort of infinite number of potential mechanisms and interactions, it does make it important that we are actually trying to measure responses and that we ask for solid evidence before sort of jumping on one of these trains. What makes this difficult from an evidence standpoint is that this points to a number of ways that nutrition really could be responsive to certain situations. So we could have suboptimal nutrition, but we may not see anything except during challenges. And furthermore, if you have a deficiency, it may not really show itself for years. So it does kind of make it feel like we're shooting in the dark a little bit. So how can we deal with that? So I'm just going to walk through a little bit of what I think we should be looking at from a research standpoint to enable us to use these insights. One, I think we can use challenge models. I'll just walk through a couple examples. So this is using a yeast product where in cows fed a diet with dry rolled corn, sort of a modest level of rumen fermentable starch. They didn't see any response to that yeast. And yet then when they hit them with a high moisture corn challenge to probably 50% increase the rumen fermentability of that starch, the control animal showed a drop in fat corrected milk with some milk fat depression, whereas those fed this yeast product did not have that drop. So again, the benefit really only appeared in that challenge. Oops. Okay. An example from the feedlot world, feedlot cattle challenged with endotoxin or LPS that were fed chromium showed the same dry matter intake as control animals, but lost far less body weight. So rather than losing 3.5 kilos per day for the window of time right after that challenge, they only lost about 1.5 kilos per day. And they then saw similar responses to chromium in receiving cattle, again, the high stress cattle in the feedlot world. Later on, they also fed it later in the feeding period and really didn't see much response there. So again, unique to that challenge window. And a third example in sheep here, sheep fed a really high level of selenium and vitamin E to stoke up antioxidant levels during a baseline period with no heat stress, really no performance benefit whatsoever, and yet during a heat stress challenge, massive improvements in ability to handle that heat load, and completely prevented the typical drop in feed intake with heat stress. And long term, we've really seen this become more and more of a focus in the dairy nutrition world. So this is just a graph showing the proportion of published peer-reviewed dairy nutrition studies that incorporated some kind of challenge. And I'm including transition cows as a challenge model here. And you can see that probably today we're up around 40% of all the dairy nutrition literature published is using some kind of challenge, making the point that we're past the point of just simply trying to dilute maintenance. We're looking for ways to use nutrition to make animals more resilient to these challenge windows. And I would argue that's the path to continued gains in efficiency with this paradigm, is that we not only focus on diluting maintenance, we focus on those big losses in efficiency that come with sick and God forbid, dead animals that don't tend to come into those calculations where we only look at healthy animals in our herds. Okay. Two, for some of these, kind of pie in the sky things, large scale research with way bigger numbers of animals included gives us opportunities to get at things that we really couldn't have a generation ago. And so, the very large farms, good or bad, that we have today give us opportunities to do research that people could not have done 30, 40 years ago. And just one example, not necessarily from a huge farm, but this is a study we just published looking at a plant called Chinese skullcap or Scutellaria bicalensis, if you're a scientist. It's a plant that's very high in flavonoids. It's a traditional Chinese herb. We went to a commercial farm and used an extract of this plant in some treatment pellets and otherwise identical control pellets. And working with a farm that's milking 700 cows on robotic milkers, we were able to use the robots to deliver treatments in a randomized fashion across 41 cows per treatment. And by going to this commercial farm, our labor costs were far less than if we'd done this on the university dairy. And with about 6 to 8 months of effort on this, we were able to show that feeding this polyphenol, this flavonoid extract, gave us a 13% increase in whole lactation milk yield by feeding this for just the first 60 days of lactation. So the dotted line here at 6.5 weeks into lactation shows where we stopped feeding that, and you can see that throughout the rest of the lactation, we continued to show a significant increase in milk yield. Okay. We don't know feed intake here. There's some trade-offs of doing this on a commercial farm, but again, the total cost of doing this and the speed with which we could do this was just much, much better than doing this on a traditional research farm. And a third approach we can take to this is to use meta-analysis. So rather than relying on one study to give us an answer, here's an example of a graph using maybe 15 studies to try to get at what's the overall effect of feeding X. But just as important, what a lot of people don't know about meta-analysis is you can also use it to get at, is there some hidden data out there that people are not publishing? We can at least get some evidence as to whether that's true or not. And we can also dig into whether there's variance across studies. And to me, this might be the most powerful part here. So when there's all these potential interactions and the possibility that a feed additive might only work in one physiological state or another, or one breed or another, we can use this approach to say, is there differences in responses across studies that then we can maybe try to attribute to physiological state or breed or what have you. And I think that can lead to controlled experiments to sort of try to verify those things that might be really useful. Okay, so to wrap up here, first take-home point, we have a lot of physiological responses to nutrients now that are largely independent of their roles as cofactors or substrates, And that just really expands the scope of what we can do with nutrients. Number two, partly because of that, I think we need to consider more maybe decision tree nutrition approaches rather than just saying, okay, here's the requirements for this animal, giving this much milk. We're just going to formulate a diet and let it ride until we, have to incorporate a new forage or the prices change dramatically. We should be thinking, in my mind, much more creatively about how do we address heat stress, how do we address an outbreak of diarrhea in the herd and that sort of thing. We don't even feed cows differently if they're in the sick pen. I think that's a really low-hanging fruit here. Number three, I think if we're going to really get at some of these ideas and really test them properly, we need a mixture of many different types of research, mechanistic studies, powerful field studies, and we need a lot of people with different expertise engaged. And lastly, what gives me hope is some of the technologies we have on cows, on feeding systems, might give us opportunities to accomplish this without becoming absurd in terms of budgets on research. And that's all I have. So I'd be happy to take any questions that anybody out there would like to throw at me. Thanks for your attention.
Well, thank you, Barry. Appreciate that presentation. If anybody has any questions, we've got a few minutes, and you can unmute yourself and feel free to pipe up with any burning questions. Any? Okay. Well, maybe while somebody's formulating one, so your Chinese skullcap research was really intriguing, and thinking about kind of these class of polyphenols, which may serve as antioxidants or reducing inflammation. Do you think really the milk response comes from nutrients that are spared having to deal with the inflammation or oxidative stress, and so those nutrients can then be put to more productive uses?
Yeah, that's a good question, Charlie. I think that's a very legitimate possibility, and that's one a lot of people talk about, and I wouldn't discount that. One of the few mechanistic insights we had in that study is we also saw a decrease in somatic cell count really through that entire feeding period when we gave that product. Mm-hmm. And that could decrease energy burn going to sort of immune activation related to mammary inflammation. But I'd also make the point that even from cell culture work, we know that inducing inflammation in a mammary epithelial cell actually changes, it changes the DNA of the cell, making it harder for it to produce milk synthesis components. And so even if you don't have a shortage of energy, which you wouldn't in cell culture, you see a decrease in milk component synthesis simply because the inflammation directly suppresses those transcriptional programs. So it could be a both and, I would say.
Okay. Very interesting. Anyone else on the call like to toss out a couple of questions? Because I could go for hours.
As could I, obviously, since I went too long.
I don't want to hog the floor either.
Good morning, Barry. This is Joe Daniels. Hey, just had a question. Can you speak to maybe what you're working on now, or anything else that may be intriguing to you that you didn't speak about this morning?
Sure. Well, I mean, I think we're very interested in the class of flavonoids. There are many molecules there, and I think one of the questions we're trying to get at is to what extent are these nutrients acting through epigenetics? So I'll back up a little bit, and probably the most interesting finding I've ever had in my scientific career is we've treated postpartum dairy cows with anti-inflammatory drugs like sodium salicylate or like meloxicam for just a 3-day window, and we've seen those cows make more milk 300 days later. And just even coming up with a plausible mechanism of how that could work is rather difficult. And so we've been really interested in starting to really dig into, is this actually changing the epigenetic programming of DNA in a way that doesn't get rewritten then for that whole lactation? So a lot of people think about epigenetics cross generations, which is maybe the most crazy application of it, but parts of our DNA, the methylation is rewritten, especially with a big event like giving birth. And we think that that window of time right after a cow gives birth, at least the mammary gland, is really open to suggestion, if you will, in terms of nutrients influencing how it rewrites the methylation then, and maybe then that it kind of sets the program for the whole lactation. So I'm excited. I mean, again, the questions are kind of limitless then, but what are all the effects we could have in that first week of lactation that we haven't dug into enough?
Great. And so just to follow on that, some of the work we did with you recently on medium chain fatty acids and inflammation, let's just say we could get them past the rumen. Do you think that could be another class of these compounds which could be used to lower inflammatory activity and perhaps, as you say, rewrite the milk synthetic machine for the subsequent lactation?
Yeah, potentially for sure. So we've been working with you guys using a cell culture model that we've kind of adapted from other people, where we're using macrophages as a certain type of immune cell to just assess whether different nutrients have pro-inflammatory or anti-inflammatory or protective effects in the presence of endotoxin, for example. And yeah, we've seen some really nice responses to those medium chain fatty acids influencing or having anti-inflammatory effects and sort of protective effects against endotoxin influence. And so assuming, like you said, you could get those to the right organs, the right tissues, and they have those sort of anti-inflammatory effects. Well, now, again, you've opened up multiple potential mechanisms. You know, maybe those fatty acids can directly influence mammary epithelial cells, or maybe they just have a secondary effect by preventing inflammatory responses to other signals, and thereby preventing sort of a damaging effect of an inflammatory event secondarily. So yeah, I mean, it's exciting stuff.
Great. Well, it's a little after 8:30 here, 7:30 where you are, Barry. So I really appreciate that discussion, but we ought to wrap it up and let everybody get out and get their day's work done. So if anybody in the audience has other questions, you can see Barry's email address up there, or we will follow up with contact information in an email to you shortly after the webinar. I would also encourage you to please stop by and visit us at the Tri-State Nutrition exhibit, and we thank you for signing in this morning and wish you a great day.